This is a first-in-class molecule that attacks cancer stemness. Specifically, it targets STAT3 phosphorylation. And STAT3 is a central role in several different aspects of cancer generation. And it's important in this cancer stemness property. So in the lab, what happens is that if you take a cancer, you can make organoids, small little organoids, and with the 608, these organoids don't develop, and so that's an indicator that you're inhibiting cancer stemness. So the study, the BRIGHTER study, really began with some preclinical data that suggested that this BBI-608 compound inhibits cancer stemness, so it's a new molecule, a new mechanism of action, and in the clinic there was some very encouraging initial activity. So that led to the genesis of the BRIGHTER trial. So the BRIGHTER trial is specifically for gastric cancer, advanced gastric cancer in the metastatic setting that has progressed on first-line chemotherapy. So most patients will have metastatic disease, have progressed on a platinum 5FU-based regimen, and then they're eligible for second-line chemotherapy. And it's a randomized phase 3 study of Taxol versus Taxol plus BBI-608. It's a placebo-controlled double-blind study, so everybody gets, BBI-608 is a pill that you take. You take about 6 pills twice a day, and the placebo arm gets the same pills, but without the active drug. And the primary endpoint is overall survival. Patients who receive taxanes in the adjuvant setting, like neoadjuvant chemotherapy with radiation, they can go on the study if they had disease progression more than 6 months after their initial chemotherapy. The study is ongoing. So this is a phase 3 study. We hope to finish accrual in the fall. So we've accrued about 2/3 of the patients, and we hope to have results about a year after the study finishes. This is the main registration study for this compound, but they have seen activity across many different cancers that exhibit a cancer stemness phenotype. There was an earlier study in colon cancer that, although overall negative, in a specific population that had a positive biomarker, was very active. So this study is, we're hoping that in all comers, patients will have a benefit, but we're also looking at the biomarker, which is the expression of phospho-STAT3. So I think that the future of BBI-608 will depend in part on this study, but in part on the interaction with the immune system as well. So one of the aspects of STAT3 is it affects the immune infiltrate. So we think that there might be an ability to partner this drug with the checkpoint inhibitors or other immuno-oncology drugs, so that we can even have better benefit. And it would be an amazing time if we could have a non-chemotherapy regimen for either first or second line therapy in any of the GI cancers.